Hi everyone, welcome back. We have reached a major milestone in our bioinformatics journey. Over the last few videos, we mastered BioPython to handle raw DNA sequences, and we also learned Pandas to manipulate clinical spreadsheets. But in the real world, these two things don't live in isolation. If you want to run statistical tests on your sequences, plot their properties, or eventually feed them into like a machine learning model, you cannot just hand the computer a raw FASTA file. You will have to extract the features and put them into a table. So today we are going to bridge that gap. So let's directly jump into VS Code. First, we will import our essential libraries, which are CQIO from BioPython, our GC fraction calculator, and obviously Pandas. So yeah, import Pandas SPD from Bio. We will import sequence IO and from bio dot sequence yeah utils. We will import GC fraction. Okay. Uh, Also, I'm going to generate a quick small FASTA file so we all have the exact same data to work with if you are working hands-on along with me in this video. So yeah, this is the file, dummy FASTA. Okay. Now we will read this file with open FASTA. We will save it. So we will put it in write mode as f dot write dummy FASTA. So we can also add a simple print statement, which will tell us that sample FASTA created. Okay, let's run this, and here you have it. What is the name of the file? Sample gene. So it should be here. This is the sequence file that we have generated. So we now have our four tiny sequences. Sequence 2 looks very GC rich, as you can see, while sequence 3 looks very AT rich. And so, yeah, let's write the code to quantify this. So the strategy here is very simple. We are going to create an empty Python list. Then we will use our BioPython iterator to read the FASTA file one sequence at a time. For every sequence, we calculate its properties. These are called features in data science and store them in a dictionary. And finally, we append that dictionary to our list, right? So let's begin. Data list will be a blank array. Then we will iterate through the FASTA file. So for record in sequence io dot parse, you might remember these commands from our previous videos, right? Sample genes dot FASTA, and we will open it as a FASTA format. Okay, next sequence ID will be record.id. We have done all these things in the previous video, so I do hope I don't have to explain these things again. Sequence length is equal to length record.sequence. Now we will calculate the GC content. This also we have done previously. GC content is equal to GC fraction record.sequence into 100. Okay, now we will create a dictionary for this specific sequence. So sequence, let's call it sequence features. Okay, after that we will do yeah, ID is equal to sequence ID. Then length is not is equal to, yeah, length is sequence length, and finally GC percent is rounding the values GC count to two decimals. Okay, and finally we will add it to our master list. So data list dot append sequence features. Yeah, that should do it. And finally, we will print a statement which will tell us extracted features for. Uh, we will calculate the length of the list, and after that, sequences. Right, that should do it. So if you were to print data list right now, it would just look like a standard Python output. It works, but it's hard to analyze. So let's first run this code. So we're not printing it right now, but if you would like to see it, we can see it like this. See, it looks like this. It just looks like a standard Python output, but it's hard to analyze if you think about it. So that is where the magic step will come in, right? So let's begin. Because we structured our data as a list of dictionaries, Pandas know exactly what to do with it. The keys of the dictionary become the column headers and the values become the rows. So yeah, let's see how it is done. df is equal to pd dot data, sorry, that will be a capital D, data frame. You need to make sure that you are correctly typing the capital and small letters because, yeah, it is case sensitive sometimes. So display df and see, there we have it. We have translated raw biological text into a clean mathematical data set. We can clearly see sequence 2 is at 100% GC and sequence 3 is at 0%. Our sequence data is now fully integrated into the data science ecosystem. So why go through this trouble? Because once your DNA is in a Pandas data frame, you unlock incredible analytic power. So we want to know, like suppose you want to know the average GC content of all your genes, just one line of code. You don't need this part actually. This is just for printing, but the main code is basically uh, here, df, and then we will write GC%, and all you have to do is just calculate the mean. We can add a percent over here. That's all. Let's see. Average GC percent 45. Right. Now, want to filter out sequences that are too short or find only the high GC sequences? Again, it will be just one line of code. We can write high GC genes is equal to df. Uh, and I think, yeah, once more we can write yeah, GC%, and then whatever is greater than 45.0, and then we can display it as well, and there you have it. Only sequence 2 has GC% higher than 45%, so we only got that as the output. Now, if you had a file with 50,000 sequences, doing this manually would be impossible. But with Pandas, it takes milliseconds. But yeah, it depends on your RAM. So if you're handling big files, make sure you have sufficient RAM to run Pandas, obviously, because if you are running it on a slow laptop with a very limited RAM, and if your file is extremely huge, then there might be some issues. So make sure that you have sufficient RAM. So yeah, so today we built a fundamental bridge. We extracted features from a FASTA file and structured them into a Pandas data frame. If you're interested in moving towards machine learning in genomics, this exact script is step one of your pipeline, basically. You have to turn letters into numbers before a model can learn from them. Now that we have numerical data, we need to visualize it. So in our next video, we are going to use a library called Seaborn, which will help us create publication quality statistical plots like histograms and box plot, etc., directly from our pandas data frames. So this notebook will be available on GitHub, and if you successfully converted your own FASTA file into a data frame today, please let me know in the comments. And if you need any type of help, you can ask your questions there as well. So please hit the like and subscribe button, and I'll see you in the next video.